Welcome aboard, fellow enthusiasts, as we embark on another incredible video. Thank you for joining us today as we dive into Nudivirus. Nudiviruses are a genus of anthropod viruses that constitute the family Nudiviridae. Insects and marine crustaceans serve as natural hosts to this family of viruses. Nudiviruses are double-stranded DNA viruses, with their genome notably ranging from 130 to 140 kilobases in length. There are 20 species in this family, assigned to 4 genera. Diseases associated with this family include death in larvae and chronic disease in adults. Brace yourself for an enlightening exploration of taxonomy as we dive into its profound implications. The family Nudiviridae contains the following genera: for Nudivirus, Beta Nudivirus, Delta Nudivirus, Gamma Nudivirus. The spotlight now falls on classification history as we delve deeper into its details. During their first discovery, Nudiviruses were classified as a subgroup in the Bacilliviridae family. Originally, it was assumed that Nudiviruses were nucleolided Bacillivirices. In 2007, the genus Nudivirus was proposed to include viruses similar to the Oryctis rhinoceros virus. In 2013, Nudiviruses were classified as the family Nudiviridae. Nudiviruses are assigned to the class Nudiviridae that contains 3 other large Nudivirus families: Bacilliviridae, Heterosaviridae, and Numiviridae. Moving ahead, let's uncover the hidden gems within species and discover their significance. Nudiviruses infect only insects and marine crustaceans. Drosophila inhabila Nudivirus Drosophila inhabila Dipterogryllus bimaculatus Nudivirus black cricket Telogryllus commodus heliathus Zea Nudivirus 1 cotton bollworm Helicobacter Zea Nudivirus 2 formerly known as cotton bollworm Homarus gamarus Nudivirus European lobster Homarus gamarus Oryctis rhinoceros Nudivirus on rhinoceros beetle Oryctis rhinoceros penis menodon Nudivirus black tiger shrimp Dichorogomarus haemobiphys Nudivirus an amphipod crustacean Dichorogomarus haemobiphys Menet mercenarian Nudivirus florinstone crab Callinetis sapidus Nudivirus blue crab Dibrotica undisimpenta howdy Nudivirus dune spotted cucumber beetle Tomalisovirus TV Drosophila melanogaster osmia cornuta Nudivirus European orchard Bicrangan Nudivirus brown shrimp Carcinus manus bacillifum Nudivirus European green crab Tipula OAC Nudivirus marsh crane fly Carcinus manus bacillifum Nudivirus Vaccinius propinquus Nudivirus northern clear water crayfish Vaccinius virulus Nudivirus viral crayfish Vaccinius russicus Nudivirus rusty crayfish. In this section, we'll be peeling back the layers of virus transmission to reveal its true essence. Transmission of Nudiviruses occurs horizontally by feeding, mating, and vertically from mother to offspring. Infections can be lethal for the larvae and can possibly reduce the fitness of the host by reducing offspring production and survival among adults. In this part of the video, we'll be diving deep into structure and life cycle and unraveling its profound impact. Nudiviruses are double-stranded DNA viruses, characterized by their rod shape. Their virions are made of a single nucleocapsid surrounded by an envelope and range from 13 to 120 in length. They replicate in the nucleus of infected host cells and in some parasitoid wasp species. A Nudivirus genome, in proviral form, is integrated into the wasp genome and produces virus-like particles called polydnaviruses that are injected into Lepidopteran larvae and are thought to facilitate parasitization of the larvae. Nudiviruses have localized infection and are associated with specific cell pathogenesis and varies among species. Vesicles containing virions have been observed for to and on and aid in infecting subsequent cells. Virions sexually transmitted during mating, like to target reproductive tissues, cause the malformation of reproductive tissues in infected adult Helicobacter. In cells, enveloped and non-enveloped particles were observed in the nucleus and have been found to successfully replicate in infected virion moth cell lines, but did not replicate in fat body cells. In ORNV, replication occurs in the nucleus in midget and fat body cells. In TMV, replication occurs in the nucleus of the salivary glands. The specific cell localization is a distinctive characteristic that differentiates Nudiviruses from Baculoviruses. 3 species form occlusion bodies OBS, TONV, NV and ORNV. INVs are transmitted orally and are sensitive to acid conditions. Host proteins may be involved in the formation of Nudiviruses. Currently, there is not a general life cycle established for Nudiviruses. Let's now shift our focus to genome and explore the ways in which it shapes our perspective. Gene content comparison and phylogenetic analyses show that Nudiviruses share 20 core genes with Baculoviruses and form a monophyletic sister group with them. Fossil calibration estimate this association arose 100 million years ago, while the last common ancestor of Nudiviruses and Baculoviruses existed approximately 312 million. Baculoviruses and Nudiviruses differ in gene content, genome organization, cytopathology, infection of adults, and most likely in host range. The 20 core genes common in both Baculoviruses and Nudiviruses are involved in RNA transcription, DNA replication, variant structural components, and many other functions. Gene content and sequence similarity suggest that the Nudiviruses CNV1 and ORNV form a monophyletic group of non-occluded double-stranded DNA viruses, which separated from the Baculovirus lineage before this radiated into dipteran, hymenopteran, and lepidopteran-specific clades of occluded Nudiviruses and granuloviruses. The coding sequences vary among the Nudivirus species and encode between 87 and 154 proteins depending on the species. The order of the genes in the genomes of the viruses found in Nudiviridae are poorly conserved. There are 32 core genes conserved among Nudiviruses that are involved in various viral functions: transcription, LIF4, LIF5, LIF8, LIF9, infectivity, PIF0, PIF1, PIF2, PIF3, PIF4, PIF5, PIF6, pachygeosymbol morphogenesis, VIP, check, FORF1, DNA replication repair recombination, NAPL, helicase, helicase 2, integrase, FEN1 nucleotide metabolism, canone function, check. The conservation of the 8 PIF proteins suggests the mode of infection for Nudiviruses is conserved. As we enter this new chapter, let's navigate the complexities of tandem direct repeat regions and unravel its multifaceted nature. Tandem direct repeats are a common feature found in Nudiviruses. These regions are characterized by their imperfect palindromic core. The number direct repeat regions vary among Nudivirus species and hypothesized to function like Baculovirus homologous regions. These regions can play a role in replication and can act as enhancers. The genes identified in various Nudivirus genomes have no homology to each other and are present in both coding and non-coding regions. ORNV 14 repeat regions 30-84 pin length with varying at. NV 14 repeat regions 11 to 42 pin length up to 96% AT rich. TONV 5 repeat regions 160 to 162 pin length. SNV1 6 repeat regions 2481 pin length. NV 7 repeat regions 58.8 toad pin length. NV 10 repeat regions. Let's now enter the realm of genome integration and discover the fascinating stories it has to tell. Some Nudiviruses have the ability to integrate into their host genome, like FENV, NENV, SENV that are permanently integrated into their host, and SNV1 that integrates as part of a latent infection. A latent SNV1 infection can be reactivated to generate a lytic viral infection. The ability of these viruses to maintain their replication after integration distinguishes them the Baculovirus ancestor. Now we shift our focus to defense mechanisms, a topic that deserves our attention. In many organisms, apoptosis can be regarded as an early defense mechanism against viral infection. Some viral genes allow the cell to survive for longer while producing more virions. Helithis Nudivirus 1 OS1 virus, a Nudivirus with a broad host range, has been shown to block an induced apoptosis gene high. A functional anti-apoptosis gene, CYP, has been found to suppress the high gene which can cause the cell to die. A second inhibitor gene ACYP to the high gene has been also discovered, but its function is still uncertain. Without wasting any more time, let's jump into the fascinating world of Nudivirus-encoded microRNAs. MicroRNAs are small non-coding RNA molecules that play important roles in the regulation of genes in eukaryotic organisms. Virus-encoded mRNAs are commonly reported in DNA viruses, and several Nudiviruses have been reported to encode mRNAs. The first reported Nudivirus-encoded mRNA was from Helichrysea nudivirus 1, which was shown to regulate virus latency. Two other viruses, Drosophila inhabila nudivirus and Oryctis rhinoceros nudivirus, have also been reported to encode mRNA molecules from transcriptomic studies. However, the role of these mRNAs and their role in virus-host interactions is yet to be experimentally determined. In this section, we'll be exploring utilization for pest control. In the Southwest Pacific Islands, ORNP has been shown to be an effective biocontrol agent against the POM pest, the coconut rhinoceros beetle. In this segment, we'll be unraveling the complexities of etymology and exploring its multifaceted nature. The word nudivirus comes from the Latin nudus, which means naked, and virus, poison. Naked refers to the fact that most do not have the dense protein bodies which surround baculoviruses. However, colluded Nudiviruses with such protein bodies, such as those of Tipula oleracea and Penis menodon, have been characterized. If you enjoyed this video, don't forget to give it a thumbs up and subscribe to my channel.